organization committee to invite me here, and thank everybody in the audience. And I will be changing the topic a little bit from the, professor's talk is about the methodological development and particularly application in the biomedical science or use model animal mice, for example. I will talk about something because I work in the veterinary school as something application as omics technology, particularly as transcriptome technology in agriculture animals. So something a little bit different from what we classically thought about the animals. In agriculture animal, particularly in the companion or wild animal, we cannot all work them in well-controlled laboratory condition, we have to think about some ecological immunological response. But the omics technology, particularly transcriptomic technology, really provides this benefit to kind of work with the agriculture animal, wild animal, because due to we are lack of this well-developed immune regions. So appreciate for that. So first, I gave acknowledge for my funding agents from USDA and from NIH pilot project. Two projects in my lab, currently in my lab right now. We try focused on this kind of, excuse me. For the agriculture animal, we use the infectious disease model, quite feel as a very pandemic viral disease. You may see it here, influenza virus and also foot and mouth disease. And in this model, we try to, with this omics technology, because it's genome-wide, we try to not only characterize the immune response, we also characterize some inter-systemic, metabolic or even neuron immune response at the interface of this kind of inter-systemic level. And another model is a mice model as obesity. It also induced by one kind of DNA virus as human adenovirus 36 serotype. They have one typical clinical sign is induced obesity. I think somebody in the audience may know that one. But anyway, as two animal models in my lab, we try to characterize this inter-systemic, even inter-organismic level response at the interface between metabolic system and immune system. Again, that's kind of integrative concepts, not only at this kind of methodological protocol level, also at conceptual level. We know the animal really sacrifices too much for our human health currently because the pandemic of this, avian influenza virus, we have been cooled for 50 million chickens already. I should remember this kind of thing, appreciate animal health. So they have a two set of transcriptomic data I try to present today to reflect this kind of systemic response we have been identified so far. The first system is we use this macrophage. Macrophage system, everybody know, this is a very plastic cell system that can be activated, polarized into different activation. So that really, that's very good cell models we can, because our targeted by different virus to cause disease, and meanwhile that differentiate into different activation status. So we try to use this cell system to determine different response, particularly immune response and a metabolic response. So you see there's different activation status. The originated from the common cells progenitor, also different. This kind of thing really as a typical system in our case for infectious disease model. Another critical thing is that cells like HIV, SARS, MERS include other disease model for ours. They're targeting cells in animal body. So again, we use this cell system for different activation status. We proceed to isolate that's a very high quality RNA to do transcriptomic analysis. So I don't need to repeat all this protocol measures because the professor's presentation already discussed very detailedly. I simply gave you results. It's no surprise thing you see that's different, that genome-wide or family-wide involvement with the immune response, like a cytokine, There's a, the good advantage, the advantages for this omics technology, but the transcriptome, you can genome-wide quantify them. Without this kind of technology, you have to depend on the single RT-PCR assay or even this high-quality immune reagents. But in the agriculture animal, we lack of this kind of thing. So omics technology really provided this thing to see. Genome-wide, we can determine this kind of response genes in immune system, the whole family-wide, the cytokine, chemokine, their differential expression among different activation status are really critical for immune response against these viral infections. So this is cytokine, that's chemokine. You can see either the different activations for macrophage or dendritic cell, that's very critical cells in immunology. For immunologists, I believe somebody in the audience may understand that part. So this is the control of macrophage status. This is the status for anti-tumor or antiviral infection and others. There's RPS, as bacterial cell compound. That's a very critical macrophage activation status for against bacterial infection. But we are very interested in this kind of antivirus activation status induced by type 1 interferons. Interferon, as antiviral or anti-tumor cytokine. So based on this one, for the typical procedure for transcriptomic analysis, not only you determine by the RNA-seq protocol, you also confirm by different classic technology like with RT-PCR. So similar kind of thing. This is a group of genes for interferon regulatory factor. As I told you, we're interested in this antivirus activation status. So we characterize this group of interferon regulatory factor. Then we confirm our RNA-seq data. At the level, not only RNA-seq genome-wide level, we will confirm it as a gene family-wide level with RT-PCR assay. They're consistent very, very well. So meanwhile, as I said, the advantage of this transcriptomic analysis is not only determine the genes in immunological pathway, we also identify such significant pathways related, like here, different transcription factor that can very basic, essential underlying the other, not only immune responsive, also development or metabolics. There's several group of factors that's very essential, very critical for the metabolic pathway. So again, a similar kind of thing, we not only characterize them at genome-wide with RNA-seq technology, we also confirmed our observations with the proteomics technology. We confirm this group of the genes we choose about 16. This kind of transcription factor. They're very, they're kind of sometimes is a very rare copied genes. We not only confirm that at the gene level, also we confirm at the protein level to see most of them genes are consistent at not only protein level, also gene level, but they do have some inconsistency between the protein level and gene level. About 1/3 of the genes, you see higher upregulation at the RNA level, but there's no really upregulation significantly at the protein level. So I will, make you pay attention on that part. So as I said, this set of RNA-seq data will not only determine the gene upregulation or downregulation with immune responsive genes. So there's pathways we determined a kind of this most related to the lipids metabolic pathway. For example, this is the pathways as in the cell, all the cell organism, there's an MPK regulated pathway that are very critical, regulators for the lipids metabolism. You can see different kind of lipids metabolism, cholesterol, phospholipids, or, I don't want to mention very detailed, but you see this one. We do want to use this transcriptomics technology to providing some interface response, not only traditional for this immune response or for the biochemicals metabolic response, we see their interface response too. So again, you know that when we profile different activation status, as a typical interferon activation status, the others as response to the parasitic infection or the bacterial infection, we compare them, we can classically get their most significant response in different pathways. So again, not only characterize the gene level with transcriptomic study, we also characterize at the protein level with this, proteomics procedure or with a traditional RT-PCR assay. We also confirmed our significant genes identification using pharmaceutical way. Like this way, I thought several lipid metabolic pathways have been identified by our transcriptomic assay. We know that for lipids metabolics, they have so many drugs identified either lipid metabolic activator or inhibitor. We have been selected 50 of this kind of inhibitor or activator. Finally, we show one kind of this lipids inhibitor, that's TOF. This is the inhibitor for this lipid metabolic pathway enzyme. That's an inhibitor of acetyl-CoA carboxylase. This is regulator for lipid metabolism, but we can use this inhibitor. You can see this fluorescence, red fluorescence. That's our virus infected cells. This virus have been labeled by the red fluorescent protein. So you can see treated with this physiological condition for this kind of lipid inhibitor, you can suppress viral infection. Something that's very interesting for us. So basically, you can see there's suppression for this. That's primary macrophages for pigs. You can suppression about 90% just by regulate lipid metabolism instead of from immune pathway. So that means this kind of in vivo, this kind of pathway, different systemic pathway are really interaction during infectious disease. So we are not only focused on immune response animal. So again, this is a flow cytometer assay with 10,000 cells. You can see a suppression of viral infection. That's in this typical cell line suppression 93%. Even in primary cells, macrophage or dendritic cell, they can suppress this viral infection in their cell lines for more than 50% too. So basically, I want to tell you, regulator of lipid metabolism can be, facilitate us to suppression viral infection. So another, there's a well-studied pathway, that's epigenetic pathway. We have not really think too much as related with regulation of infectious disease, but we do show several pathways as epigenetic, mechanism also involved with this infectious disease. Again, we confirm the different level at the protein level, at RNA level. We also with some drugs to see if they can regulate viral infection, yes or not. Yeah, quite a few of these drugs that can be regulated at the epigenetic pathway, as we revealed in the use of transcriptomic assay, that can regulate the viral infections. So I want to give you a simple summary about our characterization of this with infectious disease model. Firstly, the macrophages and dendritic cells are really very good cell line to do this kind of transcriptomic study and correlate between different intersystemic response. And secondly, several key genes, not only from immune response pathway, also from some other pathway. Typically, we don't think about them. So thirdly, with this kind of signature genes and signature pathway characterized from transcriptomics, we can use for therapeutic design, as currently we're going for vaccine design, can combine both metabolic regulation and immune response regulation. So I will very brief our second project, second data of use obesity model. That's a rat model. This model that different from the classical, we induce this obesity not only with high-fat diet, a typical factor to induce obesity, we also combined by this adenoviral infection, because in the natural occurrence of the obesity, we believe as a multi-factor observation instead of a single factor. So we induce this obesity model rise. You can see that obesity have been well induced by characterization of their adiposity index. This kind of synergistic treatments with both high-fat diets and adenoviral infection really induce more significant obesity in the model animals. So again, with this transcriptomic assay, this is toll-like receptors. That's very typical immune response receptor in all conserved in all hybrid, vertebrates. So we not only determine this kind of immune response gene. I only show you one group. Everybody knows there's so many genes, too many to show here. But there's other groups of immune response. You can see after obesity, induction of obesity, either by this high-fat diet or by this synergistic induction from high-fat diet and plus viral infection, the immune response genes are typically suppressed. This is other interferon gene response, a K-regulated pathway for interferon response. As I show you, they're significantly suppressed. I don't want to show you more about this one, but I give you one single conclusion is that immune response genes have a typical suppression during our induction of this obesity model. This is oncogene analysis and pathway analysis. I want to show you because my topic is intersystemic. I have shown you in the first set RNA data. In this set RNA data, you see that red bar is upregulated pathway significantly. That blue bar is downregulated pathway significantly. So the blue bar I will show you this kind of immune response or the metabolic. I discussed already with last set of data. But upregulated pathways are primarily related with neuron system. So that's kind of thing I want emphasized by intersystemic interaction in different disease model. So I don't want I'm because of time I don't want to extend too much, but if you have questions related with this one, I will go ahead and wrap up. So summary of this one, that's a very good model in our case, at least. So we characterization, we use this transcriptomics procedure, and not only show the immune response pathway, we also show the metabolic pathway or other even neuroimmunological interaction. So we hope that from this kind of interface or intersystemic characterization, we can really decipher some mechanism underlying these two disease models. So with this one, I want to appreciate for the colleagues involved in this work, as students and postdoc lab managers. I don't need to read it one by one. So our collaborators from our universities or from universities also, or even from England for other virus. That's typical other virus. And the funding resource, I appreciate the first slides already. So with this one, I conclude my talk and we come to your question.
Okay, we do have time for questions. I do, just to inform, we have 2 speakers this morning that are not here, so we have a little extra time now. So, we have any questions? We can do some questions. Anybody got a question? Okay, good. Here's one.
Thank you very much. I'm just wondering if you... Yeah.
Go ahead.
Thank you very much for your interesting talk. I'm just wondering if you propose a primary signaling pathway from receptors, you mentioned immunoreceptors in ileum for obesity, or you may think it is a secondary phenomena. I mean, based on gene expression.
Mm-hmm.
You think the alteration in gene regulation, in gene expression, happens first as a primary phenomena for obese people, or is it a secondary one?
I appreciate that the question is about, do you think this kind of phenomenon is upregulated at the immune pathway first or metabolic pathway first? Is that what you ask about?
Exactly right.
Yeah. So I would say, for the obesity per se, as the first slide I want to see, there should be metabolic pathways first got involved. Then the immune response, particularly the chronic inflammation things underlying got, either regulated. So after this one, the interaction between them will be, underlying this mechanism we need to focus on for obesity. That's a complex question. I cannot give single answer because we don't know. But philosophy is like this way. We need to think of immune response later than metabolic in our based model. But in infectious disease model, this kind of order is reversed. I hope you agree with me. Thank you.
Sang it again.
Thank you.
Oh, it's easy.
Oh, thank you.
So